DNA, the genetic backbone of the cell. Here we see a DNA double helix, a macromolecule made up of phosphates, sugars, and nucleotide bases. The two strands, twisted together, run antiparallel with complementary base pairs held together with hydrogen bonds. A's pairs with T's and G's pairs with C's. One strand runs from 5' to 3', while the other runs from 3' to 5'. Before mitosis, the DNA must replicate. At the origin of replication, topoisomerase begins to untwist the double helix before it unzips. Helicase, another enzyme, unzips the two strands, forming a replication bubble with two replication forks on either side. The strand running from the origin of replication to the fork is the leading strand, as nucleotides can be added directly to the 3' end of the new strand as it moves towards the fork. The strand below is the lagging strand, as the 3' end of the new strand will be in the opposite direction of the fork. The leading and lagging strands flip-flop on the other side of the bubble, but the reasoning is the same. DNA grows on the 3' end, but overall direction of replication is towards the forks. Now, let's talk about replication on the leading strand. In order for DNA polymerase III to bind, primates must first come in and add some RNA nucleotides complementary to the old strand. Now, DNA polymerase III can grab on and add DNA nucleotides on the 3' end of the new DNA strand. Simple, right? The lagging strand is a bit more complicated. First, primase adds RNA nucleotides not only on the end of the strand, but in the middle, creating gaps called Okazaki fragments. These fragments are necessary for DNA polymerase III to add on the 3' end, but still move towards the replication fork. DNA polymerase III adds nucleotides in the 3' direction in the last fragment, then repeats in the fragment closest to the fork. Next, DNA polymerase I replaces the RNA primers with DNA nucleotides. This leaves a new strand with small breaks. DNA ligase comes in to close these breaks. Both of these processes repeat, but starting on the origin of replication on the bottom strand. Notice how all new DNA continues to grow on the 3' end, but overall direction of replication and towards the replication forks. Once all nucleotides are copied, DNA polymerase I replaces the original RNA primer with DNA nucleotides, and DNA ligase closes the break. Now we have a fully replicated DNA bubble.